Cognitive and Behavioral Genetics 99. Visuospatial Construction. Department of Psychological and Brain Sciences, University of Louisville. The ability to see an object or picture as a set of parts and then to construct a replica of the original from these parts is known as visuospatial constructive cognition. Examples of visuospatial construction include drawing, buttoning shirts, constructing models, making a bed, and putting together furniture that arrives unassembled. Visuospatial construction is a central cognitive ability. At the same time, there are enormous individual differences among people in their ability to perform visuospatial constructive tasks. Some individuals draw extremely well, others cannot draw at all. Some people can copy complex patterns accurately and rapidly. Others can copy accurately but slowly. Still, others can copy only simple patterns or none at all. The importance of visual spatial construction for everyday life, coupled with the wide range of ability shown by individuals of the same age, has led to the construction of measures of visual spatial construction on virtually every full-scale assessment of intelligence. In addition to the wide range of ability evidenced in individuals with normal intelligence, the phenotype of at least one neurodevelopmental disorder, Williams syndrome, includes a hallmark weakness in visual spatial construction. In this report, we review what is known about a possible genetic contribution to visual spatial constructive ability. The remainder of the report is divided into 4 parts. In the first, we consider, in brief, general intelligence and spatial intelligence, with a focus on individual differences in visual spatial constructive abilities of people with normal intelligence. With this information as background, the second and third sections focus on the visual spatial constructive abilities of individuals with Williams syndrome or small deletions in the Williams syndrome region. We conclude that there is a specific genetic basis for the extreme difficulties with visual spatial construction evidenced by most individuals with Williams syndrome. In the fourth part, we review behavioral genetic studies of visual constructive ability, which suggests that a substantial portion of the individual differences found among people of normal intelligence has a genetic basis. General intelligence and visual spatial constructive ability of individuals with normal intelligence. The field of psychometrics has a long tradition of characterizing basic dimensions of individual variation in the human population. Much of the focus within this field has been on intelligence. There is a wide agreement that human intelligence includes 3 components: verbal ability, non-verbal reasoning ability, and spatial ability. These components are partially separable, but are not completely independent. For example, correlations between tests of different abilities are typically approximately 0.4. Thus, people who perform well on tests of one of these components are more likely than would expected by chance to perform well on tests of the other components. People who perform poorly on one component are relatively likely to also perform poorly on the others. General intelligence is indexed by these correlations. In the first decades of this century, Spearman and Thurstone laid the framework for a controversy about G that persists to this day. Spearman argued for a model of cognition in which G was the central driving force behind covariation in specific cognitive skills. Within this framework, G may be taken to index a general ability such as executive function or speed of processing. Thurstone's opposing point of view suggested there are multiple cognitive abilities that operate independently. Within this framework, G may be considered to be derived from overlapping component processes in different domains of intellectual functioning. At the end of this article, we considered behavioral genetic evidence relevant to this controversy. Attempts to characterize the nature of the spatial ability and the tests that illuminate it have isolated 2 groups of tests that assess partially independent abilities. The more powerful group of tests, often called tests of visualization, are closely associated with ability in visual spatial construction. These tests include block design tasks, such as those on the Wechsler Intelligence Scale for Children Revised, or the Differential Ability Scales, and related tasks that require participants to compose designs from parts or to specify how an object would look after a spatial transformation, such as folding, unfolding, slicing, or rotation. Most generally, these are tests that require understanding how objects or configurations can be altered within the spatial domain. It is quite easy to vary the difficulty of these tests by varying the numbers of spatial elements or the numbers of transformations required to complete a given task. For example, the difficulty of block design or pattern construction tasks varies as a function of the number of blocks required to construct a pattern and the complexity of the design to be constructed, such as the number of solid color surfaces relative to the number of two-colored diagonally or vertically divided surfaces needed. Examination of the norms for the Wechsler Adult Intelligence Test 3 indicates a wide range of ability for individuals considered to be performing within the normal range. Adults at the bottom of the normal range typically are able to construct correctly the 2-block patterns and the simplest of the 4-block patterns. Individuals performing at the mean of the normal distribution are able to complete all of the 4-block patterns and the simplest of the 9-block patterns. Individuals at the top of the normal range are able to construct all of the 9-block patterns quickly and accurately. The second group of tests revealing differences in spatial ability is concerned with changes of orientation without changes of shape or configuration. The best known of these tests is mental rotation, in which the participant indicates whether two objects are identical except for a change of orientation. Although rotation is often a component of visual spatial construction, ability measured by rotational tests is only partially correlated with the ability measured by visual spatial constructive tasks. The rotational tasks can typically be completed more quickly and spontaneously than the constructive tasks, and they may depend more on a well-learned set of unitary spatial intuitions. Cognitive psychologists have identified 3 important components of visual spatial construction: spatial working memory, flexibility in the use of spatial reference systems that are necessary for defining spatial properties, and flexibility in the hierarchical organization of objects and configurations. Individual differences in ability for people with normal intelligence have been identified for each of these components. All these component abilities are likely to be important for success on pattern construction tasks. A common finding for spatial tests is that, as a group, males tend to score higher than females, whereas females tend to score higher on verbal tests. Some researchers have suggested that this male-female difference in spatial ability has been diminishing. A number of authors expect female-male differences in spatial ability to remain and have concluded that they are due to evolution and genetic determination of sexual differentiation. Williams syndrome. Williams syndrome is a contiguous gene disorder involving a hemizygous microdeletion of approximate 1.5 megabases of chromosome 7q11.23. More than 95% of individuals with Williams syndrome have deletions of this classic length. 16 genes have been mapped to this region. Williams syndrome is characterized by mental retardation or learning difficulties, a unique cognitive profile, an unusual personality, infantile hypercalcemia, dysmorphic facial features, and supravalvular aortic stenosis, SVAS. The cognitive profile includes relative strength in auditory rote memory, language abilities that are at or somewhat above expectation for overall level of cognitive ability, and extreme difficulty with visual spatial construction. Most individuals with Williams syndrome perform at or below the bottom of the normal range on tests of block design. To address genotype-phenotype correlations involving cognition, a quantitative specification of the Williams syndrome cognitive profile, WSCP, is important. To provide such a specification, we operationalize the WSCP as a particular pattern of subtest scores on the DAS. The specific criteria are listed in the Frangiskakis et al. And Mervis et al. Of 84 individuals with Williams syndromes tested, 74 fit the WSCP, yielding a sensitivity of 0.88. In contrast, of 56 individuals in the same IQ range, but with other disabilities or low normal intelligence, only 4 fit the WSCP, yielding a specificity of 0.93. The group of 56 individuals include 20 who had been clinically diagnosed with Williams syndrome, but were later found not to have a deletion of 7q11.23. Only 2 of the 20 fit the WSCP. Thus, Williams syndrome, as defined molecularly, is associated with an identifiable and very distinctive cognitive profile. The hallmark difficulty individuals with Williams syndrome have with visuospatial construction activities is most clearly illustrated by their performance on tasks of block design, also referred to as pattern construction. Accordingly, the DAS pattern construction subtest is a critical component of the assessment of the WSCP. In pattern construction tasks, the participant is shown a target design and is asked to reproduce this design from cubes. The cubes and examples of 2 and 4 block patterns are illustrated in Figure 1. To complete this task successfully, the participant must analyze the target pattern into the parts available on the cube, locate the correct parts on the cubes, and arrange the parts into correct configuration to match the target pattern. This process requires flexibility in moving back and forth between the local organization of the parts of the pattern and the global organization of the pattern as a whole. Bellugi and colleagues provided the first extensive discussion of the difficulty evidenced by individuals with Williams syndrome on block design tasks and other measures of visuospatial construction, and they concluded that the visuospatial constructive abilities of individuals with Williams syndrome were deviant rather than simply delayed. Bellugi et al. Reported that adolescents and young adults with Williams syndrome have extreme difficulty copying even a simple 4-block checkerboard pattern, the easiest pattern on the block design subset of the WISC-R. Most normally developing 6-year-olds complete this item correctly. Although many of the participants used the correct block surfaces, most did not maintain the overall global 2x2 arrangement of the blocks, leading to broken configurations. Bellugi et al. Argue that the visuospatial constructive problems of individuals with Williams syndromes were due to attention to the local element's parts at the expense of the global element, the object as a whole. Bellugi et al. Also noted that when individuals with Williams syndrome drew pictures of objects, they focused on parts of the objects rather than the object as a whole. This led to drawings in which object parts were scattered over the page. The finding that individuals with Williams syndrome have great difficulty with a range of visuospatial constructive tasks have been replicated repeatedly. However, recent research suggests that the difficulties evidenced by individuals with Williams syndrome are developmental, part of the normal developmental sequence of acquisition of these abilities, rather than deviant. Bertrand et al. Found that normally developing 4- and 5-year-olds often produce drawings like the bicycle in Figure 2a. Production of this type of drawing is part of the normal developmental sequence of learning to draw. Longitudinal investigations of drawing ability of individuals with Williams syndrome also fit a developmental model. Clear improvement is shown over time, often resulting in drawings in which the parts of objects are clearly integrated into a coherent whole, as illustrated by the bicycle figure in 2b. Performance on pattern construction tasks also improves significantly with age. Detailed analysis of the errors made by the individuals with Williams syndrome on the DAS pattern construction subtest indicates that children produce almost twice as many broken configurations as adults for 4-block patterns. The majority of adults did not produce any broken configurations. Furthermore, the proportion of incorrect patterns produced by adults for which the correct block surfaces were used but placed in the wrong position or orientation, the type of error closest to the correct pattern, was more than double that for the children. Finally, Pani and al. Have shown that global perceptual processing by individuals with Williams syndrome is largely normal. All these findings are consistent with a developmental rather than a deviant account of acquisition of visuospatial construction abilities by individuals with Williams syndrome. There are also clear individual differences among persons with Williams syndrome in performance on measures of visual spatial construction. For our sample of participants, standard scores of the DAS pattern construction subset at all ages range from 20, the lowest possible score, to 38, which is at the 12th percentile, definitely within the normal range. On the developmental test of visual motor interfration, standard scores range from less than 55, the lowest possible score, to 80, 9th percentile. From a qualitative perspective, some adolescents with Williams syndrome produce integrated drawings that are recognizable as the intended object. At the other extreme, some adolescents produce unrecognizable drawings composed of isolated objects parts strewn across the page. On the DAS pattern construction subset, approximately 10% of adults with Williams syndrome are able to correctly construct even the most difficult 4-block patterns, albeit very slowly. Of the remaining adults, some are able to construct simple 4-block patterns and some complex 2-block patterns. Others can construct only the simplest 2-block patterns. Performance of individuals with Williams syndrome on measures of visual spatial construction was highly correlated with performance on measures of other components of intelligence, as would be expected, given our earlier discussion of G. For example, even after we controlled for the effect of chronological age, the performance of a sample of 50 individuals with Williams syndrome on pattern construction correlated 0.46 to 0.49 with 3 measures of verbal ability, 0.48 with a measure of verbal working memory, and 0.57 with a measure of nonverbal reasoning ability. The individuals who performed best on pattern construction were those who had relatively good vocabularies, verbal working memory, and nonverbal reasoning abilities. Observation of participants during testing indicated that most tried to work their way through both the pattern construction and the nonverbal reasoning metrics problems verbally rather than trying to use a spatial strategy. Thus, participants tended to use a compensatory strategy to solve pattern construction problems, attempting to turn a spatial problem into verbal ones. The participants with the highest overall intelligence were the ones most successful with this strategy, as evidenced by their relatively good performance on pattern construction problems. For these and the remaining Williams syndrome participants, overall verbal ability and nonverbal reasoning were consistently superior to level of performance on visual constructive tasks. Individuals with small deletions of 7q11.23. Because the Williams syndrome deletion involves less than 16 genes, genotype-phenotype correlations would be extremely difficult to identify on the basis of participants who had classic Williams syndrome. Individuals with more subtle mutations involving only one or a few genes are therefore crucial to genotype-phenotype correlation studies. This strategy has implicated one of the deleted genes, elastin, in the vascular pathology associated with Williams syndrome, including SVAS, certain facial characteristics of Williams syndrome, and connective tissue problems associated with Williams syndrome, such as inguinal hernias and hoarse voice. Individuals with mutations of only elastin do not share other Williams syndrome characteristics. The strategy of studying individuals with small deletions has also been used to implicate LIM kinase 1 in the visual spatial construction difficulties associated with Williams syndrome. 2 kindreds with SVAS and small deletions of 7q11.23 were identified. Phenotypic characterization indicated that, as expected, most kindred members with deletions of SVAS, a few facial characteristics of Williams syndrome, and some of the connective tissue problems associated with Williams syndrome. As in previous studies, variability of expression and incomplete penetrance of autosomal dominant SVAS was found. Continued phenotypic characterization indicated that family members with deletions had not had infantile hypercalcemia, did not fit the Williams syndrome personality, and did not have mental retardation. Performance of affected members on measures of auditory rote memory and language was similar to that of unaffected relatives. However, most affected members performed less well on measures of visual spatial construction than would have expected for their overall level of ability. These individuals fit the WSCP. None of the affected members shared any Williams syndrome characteristics, including the WSCP. All family members had IQs in the low average range. Molecular genetic characterization indicated that deletion size was approximately 84 kb in one kindred and approximately 300 kb in the other. Continued characterization indicated that the smaller deletion included only elastin and one other gene, LIM kinase 1. As indicated above, kindreds with mutations of only the elastin gene do not evidence any cognitive or personality aspects of the Williams syndrome phenotype. In fact, elastin is only negligibly expressed in the brain. In contrast, LIM kinase 1 is strongly expressed in different regions of the brain, with highest expression in the cerebral cortex. This pattern of findings indicate that hemizygous deletion of LIM kinase 1 contributes to the visuospatial constructive difficulties of individuals with Williams syndrome. We have since characterized 3 additional kindreds with small deletions and low average intelligence. Affected members of these kindreds fit the WSCP, but none of their unaffected relatives did. Members of all 5 kindreds tended to talk their way through pattern construction problems. Tesabehi et al. Have reported findings from 3 additional individuals with small deletions of 7q11.23, including both elastin and LIM kinase 1. On the basis of cognitive features of these participants, Tesabehi concluded that haploinsufficiency for LIM kinase 1 may be necessary for impaired visual spatial construction, but is not sufficient. However, all 3 individuals were of higher intelligence than members of the kindreds we studied, and 2 of the 3 had above average intelligence and excellent reasoning abilities. Given these strengths, these individuals would be expected to successfully use verbal compensatory strategies to solve pattern construction problems. Thus, not surprisingly, these individuals did not fit the WSCP, which includes limits on the absolute level of performance on DAS pattern construction consistent with mental retardation or borderline normal or low average intelligence. In summary, the hallmark cognitive weakness of individuals with Williams syndrome is visuospatial construction. Study of kindreds with small deletions of 7q11.23 indicate that hemizygous deletions of LIM kinase 1 contributes to these difficulties. Although visuospatial construction is the area of greatest difficulty for most individuals with Williams syndrome, there are still individual differences among people with Williams syndrome in level of visuospatial constructive ability, which correlate strongly with individual differences in verbal ability and in nonverbal reasoning ability. Any genetic basis for these individual differences is likely due, at least primarily, to regions of the human genome rather than 7q11.23. Behavioral genetic studies of visuospatial construction. Our research demonstrated that hemizygous deletion of LIM kinase 1 on chromosome 7 forms a foundation for the deficit in visuospatial constructive cognition evidenced in Williams syndrome. The genetic basis for normal variation in visuospatial construction, however, is less clear. Barring other evidence defining DNA variation in LIM kinase 1 and associating genetic variation with phenotypic variations is a reasonable place to begin the search for genes influencing visuospatial construction. It is possible, however, that LIM kinase 1 is necessary for the development of normal visual spatial construction, but that it does not account for a significant amount of the variation in the normal range. Regardless of the role of LIM kinase 1, the genetic underpinnings of the visual spatial construction as a complex behavioral trait are likely to follow a quantitative trait loci model described by a multi-gene system, with each gene contributing to the distribution of individual differences in visual spatial construction. Behavioral genetic research and traditional psychological research can inform molecular work concerning the genetic basis of visual spatial construction. Twin and adoption studies indicate that visual spatial construction, as measured by block design tests, is heritable with the proportion of variance accounted for by genes in the range of 0.44 to 0.68 in older children and adults. There is also evidence that the H-squared estimates of visual spatial construction increase from toddlerhood to childhood, and many include both additive and non-additive effects. Beyond estimating the heritability of visual spatial construction, behavioral genetic work draws on the rich psychological tradition that attempts to disentangle the complex phenotype concerning the relation between specific cognitive skills, such as the visual spatial construction, and general cognitive ability, as described at the beginning of this article. Current research has begun to focus on whether models such as those of Thurstone or those of Spearman better account for the genetic underpinnings of visual spatial construction. It is the case that there are multiple independent skills with a fairly simple mapping between simple genes and individual skills. Or are we more likely to find a complex system of genes that influence visual spatial construction, other specific cognitive skills, and G? Recent work with sophisticated multivariate behavioral genetic models indicates that the answer may lie somewhere between the views of Spearman and Thurstone. Analysis of genetically informative data by means of a hierarchical model provide evidence that specific cognitive skills can be accounted for in part by genetic influences that are specific to individual skills as well as common to many of them. Peterson et al., for instance, used multiple regression models coupled with a powerful twin adoption design to demonstrate that a significant amount of variation in block design scores can be attributed to genetic influence that affects block design, but not other WISC-R subtests. At the same time, however, a large portion of the variance in block design scores was accounted for by genetic influences that are shared with many other cognitive skills. In fact, consistent with purely phenotypic models, block design was found to be one of the most heavily G-loaded abilities relative to other commonly measured cognitive skills. Therefore, it appears that there are genetic influences on visual spatial construction that are unique to this cognitive skill, as well as genetic influences that are mediated by G. There is some evidence that the hierarchical model implying both unique and general genetic influences on cognitive abilities may be reflected at the molecular level. Petrel et al. Examined the relations between 8 DNA markers known to be associated with cognitive ability and the subsets of the WISC-R. They found significant associations between 2 markers, EST-083 and HLA, and the block design subtests. These 2 markers also significantly predicted scores on many other cognitive subtests included in the study, indicating that they are most likely influence a basic process central to G. In second analysis, the effects of G were removed prior to examination of associations between individual markers in block design. One marker, ADH5, significantly predicted block design scores even after Petrel et al. Accounted for the influence of G. Moreover, block design was the only WISC-R subtest to be significantly associated with this marker. Thus, normal variation in visual spatial construction is likely associated both with genes that influence cognitive ability in general and with genes whose influence is relatively specific to visual spatial constructive ability. In summary, large individual differences in visual spatial constructive ability are found among people with normal intelligence. Individual differences in visual spatial construction are also found for individuals with Williams syndrome. However, the range of these individual differences is restricted to the lower part of the normal range, or in most cases, below the bottom of the normal range. The visual constructive abilities of individuals with Williams syndrome are also substantially lower than expected on the basis of overall level of cognitive ability, suggesting specific genetic involvement in the determination of visual spatial construction. At the same time, visual spatial constructive ability is strongly correlated with both verbal ability and nonverbal reasoning ability. Studies of kindreds with small deletions have implicated hemizygous deletion of LIM kinase 1 as contributing to the difficulty that individuals with Williams syndrome have with visual spatial construction. Behavioral genetic studies have indicated that visual spatial constructive abilities have a substantial genetic component, most likely fitting a QTL model, with some genes involved in determining visual spatial constructive ability affecting cognitive ability in general, and others that are likely to be relatively specific to visual spatial construction. This QTL model likely accounts for individual differences within both the normal population and the Williams syndrome population. This is Melinda the Good here. Thank you so much for listening to this video. If you have made it all the way to the end without falling asleep, then you should really consider a career in research or talk to your doctor about your sleep disorder. If you enjoyed, please consider leaving a like because this video took a lot of energy and time to create. If you hit that subscribe button, I promise I will never do something like this again. Once again, thank you and good night.